Okay, I got it. Let's hope it won't be interrupted. Oh, cool. Yay. Again, you guys are like a test generation for this. This was never done. So we'll see how it is. I have to test it to see what's the best for students. But I know for sure that I don't want my average to be very low in finals. I think this will help. Anyway, so we are starting today with a new unit, and every unit will be harder and harder. I will say that the first one was the hardest, or the easiest one. Sorry, the easiest, the easiest one. So everything else will be harder and harder, and then when you reach unit 6, that's when people start dropping and giving me bad evaluations. I'm kidding. It's not really. It's actually, if you like biochemistry, this will be cool. So, again, for people, we will announce later on if somebody was not in class. Marisa is hosting extra lecture. So we are starting with bacterial growth unit, and mostly this will be physiology of bacterial growth. And I will start with a lecture 1 today, and I probably will start with the lecture from Thursday, simply because Marisa has her assignment 1 lecture on Thursday, and I want to make sure she has enough time next class. So in terms of bacterial growth, you probably heard many times bacterial growth. If you ever worked in the lab or if you work in eukaryotic cell culture, then you probably didn't hear a lot of this. But today we will evaluate the bacterial growth from some slightly different perspective, which will be physiological perspective. But in shortly, definition of growth literally means when one of the cells increase in volume, so increase, and then when it reaches certain volume, it splits, divides into cells. So basically, during the growth, what you will see is increase in cell number and biomass. So in case of bacteria, cell number usually means more biomass. I say usually because it's not always the case. So bacteria or microbes, I should say, I should use the word microbe because we talk about general microbes, not only bacteria, but I'm mostly in bacterial world, that's why I'm using bacteria as an example. So they're growing via binary fission. That means that cell is going through a division, and then when cell divides, you will have 2 daughter cells, and then 2 daughter cells will divide, and you will have 4 daughter cells. So that means binary fission. From one single cell, you get 2, and then the number increases proportion. So you probably already know this, but what you maybe did not hear about is what's happening in microbial cell from biochemical perspective during the growth. So from physiology standpoint, and what you have to know, and this is where we understood it during the first unit. So in order to have 2 completely formed daughter cells, so cell needs to generate new peptidoglycan, like new cell wall material, but before that, cell has to volume. So in order to grow in volume and to make a space for new peptidoglycan monomers that will incorporate autolysins, those like chewing enzymes, hydrolytic enzymes, will create openings in old cell wall. Literally, a cell is growing, those autolysins are chewing and opening the cell. And why autolysins have to make that hole? Because new monomers, and we talked about synthesis of new monomers last class, now have to incorporate and to literally fill up that gap in the hole. But first, what's happening is cell is bigger in volume, so cell is growing. That's the first thing that has to happen. Next one is, so when cell grows in volume, that has to be followed by increasing certain molecules. So what molecules that will increase, like whose number will increase, are of course RNAs, especially rRNA, like ribosomes, because ribosomes are needed for protein biosynthesis. So RNAs and proteins, and we will mention later DNA, are required for biomass increase. So all molecular biology associated molecules, DNA, RNA, and proteins, are following the increase in cell volume. Now, as I said, so RNA and proteins have to increase in amount. What is very important in growing cell is to have chromosome or DNA associated with replicating. What that means is that in order for cell to divide completely, the chromosome needs to be fully replicated, because we must ensure that all now formed daughter cells have two chromosomes ready. So when they pinch off, when both of them separate, each of them has a copy, properly formed copy of chromosome. And we'll see how that matters. So literally, this complete chromosome replication, like full division of the genetic material, is a signal for cell to form something that we call the septum or divisome or division septum. So that should not happen before all DNA is fully replicated, simply because that's the way to ensure that mother and daughter cell are equal. So again, as I was saying, so cell will grow in volume, and you probably saw this picture in the last class when we talked about FTSD protein, and there is a reason for that. That protein is actually very important for formation of division, for physiology of division. So again, cell increases in volume, chromosome divides, septum forms in between, and you can see this blue yellowish like a line with a yellow line inside is actually the division septum, and the time that passes between, like from the moment when cell starts until you have two daughter cells, doubling time is called generation time. So that generation time is the time needed for cell population to divide, to double. And there is a certain order of events that have to happen, and the picture here is actually showing that. So you have the chromosome, like circular chromosome inside of the cell, and again, the chromosome replication has to be completed. Very often you will have a plasmid that needs to divide. Once when that happens, when chromosome is fully completed, cell will elongate, like grow, and then when it's growing, when it's elongated, like stretched enough, you will have the septum form, and you will have that division, which is literally from the point A to point Z generation time, like a dot. And you probably heard about that too. Again, the septum formation is usually in the middle of cell. And in that case, you will have, when septum forms, there will be exposed cell wall material like peptidoglycans. Then autolysis that we talked about, hydrolytic enzymes will create a walls in the cell, like in the cell wall opening, basically leaving a space for new monomers. And you will get this septal ring, which is literally a stretch of proteins, like pieces of proteins that are in between two almost formed daughter cells. So once when you lose those proteins from septal ring, you get the pinching off to daughter cells. So try to imagine from 3D perspectives how that looks. It's actually, this is still quite easy. Now again, from the physiological perspective, again, the most important is, again, the most important are these proteins that are controlling cell division. This is what will come up and on exam and on your quiz. So again, generation time is something that you probably mentioned many times till now. So it is the time needed for a population to double in number. So how many minutes or hours or days is required? So that complete chromosome replication is a signal for septum formation. And when septum forms, you will get pinching of two daughter cells. But how do we know what proteins would control this septum formation? So these proteins are quite important, and they're first mean proteins that we will cover in detail. There are two of them that are important, and there is FTSZ, which is going to be replacement for mean proteins later on. And these details will be quite covered in midterm and on final exam or on quiz. Now, so you again, mean proteins are the first one that come into the physiology of cellular division and septal formation. So the first molecule that will actually lay down and form the location of septal will be this E protein or min E. So min E is what determines where the septum will form, and the decision points when min E will form is based on oscillation of proteins from one side to another. Now, you might think min E is the most important, and it is very important, but there is another one that kind of lead min E, and that will be protein called min D. So think from like DE, min DE protein. So min D is the one that kind of leads min E protein in the cell. And think about min D as something that expresses along with min E. So min E is what forms that division septum. So min E will determine once where min E is stationarized, that's where septum will form. But min D is required because it literally leads like this. So they oscillate as cell is growing. Like just try to imagine from 3D perspective. Cell is growing like this, min D expressed and literally takes that min E and leads like this. They oscillate, they dance together. And they're waiting for chromosome replication, RNA and protein synthesis, because they want to ensure that both cells, both future cells have enough material. So what is the signal for this dance between min E and min D to stop is complete chromosome replication. So once when that happens, min E, which again is led by min D, but E is the one that is the primary former of that septum in between. So min E now stationarizes in the point of septum and determines where division will occur. Well, that pinching will occur. So both of them are equally important. I would have a hard time to decide which one is more important, but later on when you see mutants, you can maybe conclude which one you would rather have. So again, when min E is in the center, that's the beginning of septum formation. Now, when min E, so you now have already min E ring formed, and this is the invitation signal for this main protein that is building septum called FTSD. This is, do you remember that we talked about FTSD when we talked about bacterial analogs to eukaryotic cytoskeleton? So FTSD is one of them. So it forms now, so it replaces min E. So FTSD is the one that actually forms final division septum. So this division is actually made by FTSD. So min E gets out. Why is FTSD important? So first of all, it builds division septum. Second of all, FTSD has ability to do hydrolysis of energy molecules. So this one can be either GTP or ATP. I'm not sure which one is more efficiently used, but both of them are energetically similar. And these are required to get an energy for constriction of the ring, so to get that pinching off. Now, at the same time, because this is all dynamic process, I don't know how to turn off notifications, sorry. Somebody will help me. By somebody, I mean, I don't know how to do that. I don't know. So basically, what's happening at the same time is cell wall monomers that you now have synthesized. And think about all that, it's happening dynamically. So cell wall monomers are synthesized and split, and you will have alkalizins that create openings, so new monomers can build up. And again, just to get the full sense of it, once when you have everything formed, so FTSD is now aligned with the hydrolysis, divisome is going to shrink and make the septum, and that will eventually lead to the pinching off of two daughter cells. once when pinching occurs, now you have two daughter cells, and now mean E and mean D can start their oscillation dancing again. Why? Because then you have a new round of division, because now those two daughter cells, each of them will divide in two, and you will have four of them. There are a few additional proteins that help in FTSZ divisome forming. Again, FTSZ is the one that forms divisome. Min E is preceding it. Min D leads min E. But this protein ZIP-A is just an anchor for FTSZ. And there are another divisome proteins that I usually don't really ask. They're FTSKIA. Most important for you guys is to memorize FTSZ and all physiology behind FTSZ, min E and min D. And I will again emphasize FTSZ roles are very important, and we already talked about why FTSZ is important. First of all, it's housekeeping gene in cell, so it's essential. What that means is that if you want to delete it, if you want to knock it out, you can't. You have to make something called temperature sensitive mutant, and because if you just knock it out, cell cannot grow, and you cannot examine something that doesn't grow. So that's why we are creating something called temperature sensitive mutants of FTSZ. So what usually scientists do in this reason, they create a different version of FTSZ, and that's literally missense mutation. So it's not nonsense mutation, it's a missense mutation. So it's like a change the function of FTSZ, like not change the function, but alternative version of FTSZ. So you just alter like a base pair, usually one base pair. So the gene will produce a slightly different version of FTSZ. It will have like a different amino acid structure. It will be like altered function of FTSZ. What that means is that it's standard permissive temperature, like because it's a temperature sensitive mutant, so usually low temperature. FTSZ will have the function of essential gene, and in non-permissive temperature, FTSZ lacks the function and grows with a long filament, which is a mutation, and eventually that leads to cell death. But you can examine those long filaments later on. So that temperature sensitivity, like that standard versus permissive, non-permissive temperature, actually allows scientists to examine FTSZ function, because if you just knock it out, you can't get cellular growth. Like this, you will get a growth, but it will be slightly, well, not slightly, very different morphology. So again, in terms of FTSZ, we talked about its analogy with cytoskeleton. So it's going to be analog to the tubulin. Which one is actin analog? MRE. Yep. Am I right? Yes. Yeah. Okay. And which one is intermediate filament analog? Juvenils. Yeah, because it goes all around. Yeah, it goes all through the cell. It connects everything. That's why it's called intermediate filament. That's how I remember it. So again, tubulin homolog. It's the main protein that forms septum. So don't get confused. Min E is not the protein that forms the septum. Min E is a very transient. It's required, but it's not the main. And again, I'm telling you this because it comes as a question. So septum created by FTSZ, but septum control is done by min E. So, and you will see later on why. So, so FTSZ is really, really highly significant because controls most of the protein's contractions, and it allows pinching of two daughter cells. So how is the septum control done by min E? So this is this action performance which we are going to cover. So who oscillates first? What do you think? Min D. So min D is the leader. So if you think about the dance, because they are dancing, they're like a dancing partners, min D is the one that leads the dance. So min D invites min E for a dancing. So min D has to oscillate first. So min D is important because it prevents septation, early septation. It prevents septation before chromosome is not fully replicated. And it's important that for cell to has enough material, most important chromosome replication, but also RNA and proteins. So again, min D will oscillate back and forth in elongating cell, and it will inhibit, and that min E lead will inhibit early septum formation. So it min D will literally prevent min E to form early septum before you have enough, before cell has enough material to complete both cells. That's so the one who controls it is min D. So basically min D is the controller of the septum formation, observes when all molecules will be formed, and once when it is formed, it releases min E to form early septum, and then FTSE comes. Actually, ZPA and FTSE. So again, min E will be there. So I usually imagine that as min D is holding that min E. That's why I associated with a dance. So min E oscillates along min D, and min E can actually form that septum only when min D inhibition is removed. And then min E gets replaced with FTSE. So the signal for min E ring formation, so signal for min D to stop and to release min E is complete chromosome replication. And then there are other proteins that come. FTSE is the most important, but you have that ZPA that's an anchor for FTSE. So the way how you should imagine this is that min E is very transient, and the FTSE proteins are like a scaffolding on the this is very simplified. All division process is way more complicated, and if somebody wants more details, just let us know, and I will post the detailed explanation question for it. Once when ring FTSE is complete, then min E ring can disintegrate. And one of the interesting things that I really like to ask are those min E and min D mutations, which again, you can, try to memorize, maybe picture, maybe just explanation. So what will happen in wild type? So in wild type, which is the red, min D will oscillate from one side to another, and then you will see that formation of septum in the middle, which is like a middle. In min E mutant, which is the green one, so min E mutants, there is no min E, so there is nothing to form early septum. So that means that min D is all around the cell, and you will get formation. You will not get, you will see there will be no septum. So if you see the green picture, so there is nothing to form septum. So min D is getting crazy all around the cell. FTSE mutant, which is that temperature sensitive mutant. So think about as altered form of FTSE. So it's not completely knocked out, it's altered form. So you will see multiple min E rings forming, and then min D will literally oscillate between each min E segment. So it's uncontrolled oscillation, and you see this filamentous type of the cell. So and again, these are some videos to what that not really sure they didn't work in my computer, but I realized that this morning. But if it doesn't work, shoot us an email or post on forum, and I will find the better. Any questions so far?
Maybe at the end, when you always just populate the cell until they have that moment after the chromosomes are replicated, then they go from free.
So I would, let's see.
They're just generally oscillating.
Since they're oscillating all the time, but they form the septum once when cell has enough material to divide. So they're always present. Yeah. I would say mean deletes mean oscillation. So they are controlled oscillation. If that makes sense. So I'm just very careful about how I will say it, so I don't say something incorrect. They are there, but mean deletes mean AE. They're not just going around like crazy. They're very organized oscillation. Make sense? Okay. And thanks for the question. So how do we know how many cells we have? How we quantify bacterial growth? And many of these things will sound like repetition for you because you probably heard about all this. But again, when we start next lecture, it might be a little more complicated. So how we quantify bacterial growth? So to understand that, you have to know the mechanism how to count bacterial population. Can somebody tell me some way to count bacterial growth? How you know that your bacterial cell grew? I just said colony count, great. The other, did you ever do OD? Optical density? Yeah, I personally hate it. It's completely inaccurate and I cannot stand doing it. I do it all the time, which is the reason why I don't like it. I like other way of counting, and I will tell you why it doesn't matter. Why it matters which type of the growth determination you're doing. So there are a few mechanisms to count bacterial population. So you can count by microscopy, which Katja said, which means you use a microscope. So you have a light microscope, you take 10 microliter of bacterial culture, you put it in the slide, you cover with a cover slip. You take that device that I don't know how to say in English, but you press it and you count how many cells you have. You go, 500 in, I don't know, 10 microliters of cell, and then you count how many colonies, colony-forming units, or CFU, you have per milliliter of cell. Very often you have to do dilution because bacteria grow very robustly. So you multiply that with dilution factor, and you can say I have like a 1.8 times 10 to the 9 cells per milliliter. Another way to count bacterial population is a viable count. This is actually that viable count that I just described. So this is something that we do commonly, but the question there is how many cells are really alive. You can do like that staining to and count that. And again, the problem with that viable count is usually in viable count, you will do serial dilution. So you will have like 10 to the minus second, 10 to the minus third, fifth, I know 10. And then you will just let it count until the growth is formed. So for that, so this is what I usually ask my students to do. Like I would make them to grow bacterial count with the lab. 0 hours they will take over here. Will come and they will at the same time they will like 10 microliters on the plate, and they will grow within the span. So if you took 391, this is what we did in 391. So and then you you count how many cells. The problem is that with this one that you actually have to know exact media that bacteria are using to grow. Because for some clinical isolates like pathogens, this is well understood and often done. So you can do it very easily. You probably did that in Micro 265. Some of the organisms, you don't actually know whether LB agar is the right media for it. So you might think that you don't have enough cells, but actually you just didn't find the right media for that. So that's why it's a little key. So both of these, microscopy and the viable count, are something that we call direct measure of microbial growth, which means you literally see either under the microscope or on the plate after 24 hours, actually 18 hours, you see how many cells you have. This might sound as very accurate, but it actually really depends on your pipetting technique. And as I said, for viable count, what if you just didn't get the right media? Another thing that we actually do very commonly in the lab, and in all labs you will actually see that really often, is optical density, which means you are measuring absorbance at specific wavelength. So this is something that I do all the time. You measure absorbance or optical density at 600 mostly for E. coli. For methanotrophs, which we use, or methylotrophs, which we use in the lab, it's 540. Those are standard, 600, 540, you can go with 540. Where is the problem? So for the way how you do it, you take a milliliter of your liquid culture, you put it in that, and you put it in spectrophotometer, and then the ray goes through it, and literally it's based on the density of cell population. Where is the problem? So this is, I mean, this is just a figure that you might see, and this is what we used to do in the lab, and that's the reason why I ask my students when I'm training them to do like a first optical density, and at the same time they put something on the plate to do variable count of column-forming units, and then we form something like mimicking a standard curve. So on x-axis we put optical density. Let's say at 0 hours it's going to be 0.01. And then on y-axis you have how many columns you count after that. And every hour you do optical density and you count number of cells and you put OG at y-axis, CFU on, actually OG on x-axis, CFU on y-axis, and then you form like a linear growth. And then what you can do, and we did that in class, we will get like an equation, literally for standard curve. And in that case, you don't have to do cell count ever again. You just implement that in equation because you will measure your OD you will put as X and you will get Y. So you don't have to do cell count. Or you just get undergrad, which for you because you don't like. Which I do. I really don't like measuring counting cells. I like to do something different, and let me first tell you why what's the problem with OD. And again, this is something that I encountered in my life, and very obvious problem. So when you're doing optical density, what that means is literally you are taking a mill of your culture and you're putting in the cuvette, and then you put that in spectrophotometer and you shine a ray. Specifically. What that means? That means that whatever you have in that culture, no matter, so we claim that it's based on cellular density. Well, it is. But what can happen, again, this is real life experience, what can happen is that your cells maybe don't grow. They maybe produce something that's yellowish, something like that. And that yellowish thing is actually product of your live cells because carbon source is toxic. But nobody grow on carbon source, they actually die. But you measure OD, you actually just measure the change in wavelength absorbance, and you can think they grow. Because this is indirect measure. You don't actually know whether that change originates from the density or from something. So that's the reason why, again, it happened to me that I had a false positive results and I got really depressed. So that's when I really decided I will never do it again unless realized. And I prefer this dry weight measurement, even though it's indirect, like you don't really see cells, like you just measure biomass. The reason why I like it is because it gives you actual data of the biomass that you have in your, like on your plate, or actually in your culture. The way how you do that is you take the known volume of a culture, let's say 15 mils, culture will grow in, I don't know, 5 days, you spin those 50 mils and you resuspend in certain amount of water, usually we do like 5 to 10 mil. You take aluminum dish, you measure empty aluminum dish, you record that weight, you pour your resuspended cultures, like 15 mils, for example, and you let it dry. Depending on the temperature, if you are very impatient like I am, you put it at 7 days because you really want to finish that, and then you remeasure. So that difference between empty dish and your biomass is what is your growth. So for me, this is the most accurate, I mean, again, not the most accurate, but the one that I prefer, simply because in the cell count, even though we say that there's direct measure, and it is, it will really depend on your pipetting technique. It will depend on your cells. What if your cells really got stuck together or they have very tight contacts? So you actually have way more cells that you actually counted in your microscopic slide. So there are all different problems associated with that. Another thing that you probably can do, and again, it's not really so common, at least not in our labs, but it's precise, it's a protein determination. Because if you remember at the beginning, I said that when cell is growing, you will have increase in RNA and proteins, which means it will, like you will get more proteins means more growth. So you can do a protein determination assay, or you can do any of the molecules. Most commonly is protein determination. It's done by Bradford. It's done by Bradford assay, where you measure again absorbance at different wavelengths. Another thing that is very usual in microbiology, especially if you grow microbes that are not really high in biomass, which means they even not ordinary biomass, is measuring the product at the end. So it's easier to measure biomass if you have E. coli, or if you have fast-growing methanotrophs like methylmicrobial malton BHG. But if you are dealing with some very nasty organism that just won't form biomass, it still grows, but no biomass. So what will you do? You can't really measure OD. There are no cells to change the density. So you will measure some product. You can measure carbon dioxide. Marista will tell you where is the problem with carbon dioxide. Actually, I will tell you. But so carbon dioxide is usually a result of metabolism. So whatever you oxidize, you can produce carbon dioxide. The problem with carbon dioxide as a product, which we say is a product of respiration, is if you have an organism that actually can reassimilate that carbon dioxide. So think about it. You have a carbon source 1, you oxidize to produce CO2. But then somehow your organism has a gene to assimilate back that CO2. So puts it back in biomass. So you actually don't have CO2 anymore. So you have to find another way to get product measurement. So again, it really depends on the organism that you are evaluating, what type of growth determination you could do. So if you're dealing with this CO2 issue, probably I would go with this respiration measurement with tetrazolium salt. So what is tetrazolium salt? So it's a respiration substrate. And we will learn, God, this is annoying. And we will learn later on about respiration. We will fix it later. I hate it, but fix it, please, because it really tetrazolium is respiration substrate, and what's happening during respiration, you have a set of oxidation and reductions, and one of the first steps, actually, the first step is reduction equivalent NADH that gets oxidized to NAD+. So when it gets oxidized, NADH, tetrazolium gets reduced, but you must have that NADH, which is the first step in electroplastic chain. We will cover that in unit 6. So that oxidoreduction will increase as the cell is growing, but the way how you will record it, when tetrazolium gets reduced, it changes color. So the change of color corresponds to the intensity of respiration. And this is just a picture of chemical reaction, which of course you don't have to know. One of the common things that and I will give you the example that we are using in the lab when we are measuring the growth of bacteria called ammonia oxidizing bacteria or AOB. So those organisms, again, you mentioned them later on during course, but they are using ammonia as a substrate. They oxidize ammonia for minus, but they just won't grow. Like there is no biomass. What we do, we put mineral indicator in media, so it doesn't change anything in terms of growing conditions, but as they are growing, they are making NO2 minus, and then NO2 minus will change the pH of the media, and then you tag that as a pink, because red is pinkish, right? It will change that as yellow. So if you see like very ugly yellow color, that means they're growing, and then you're happy. But if you see that they're cloudy, then you acquire contamination. So it's basically a change in pH is what is detected through change of color. And again, all these OD, dry weight, and product concentration are called indirect versus direct measures, which are cell count and viability. And again, we very commonly use indirect measurements, but very often in microbiology classes, you will see direct measurements, especially viable count. So one of the big problems that we see when we are trying to determine bacterial growth is that, again, that's another problem with viability count. We don't exactly know which media microbes will use because many of them are not cultivated. So especially when you have organisms that are in human gut or soil or marine water, so you won't see any direct count, you won't see any viable count. And that's especially a problem with marine ammonia oxidizing archaea. Like they won't grow on the plate. So they're dead. They will make NO2 minus, but you won't see them on a plate. So again, depends. You do need to know what would be your goal. So this is, so we call it, again, just an example. You don't have to know these great plate count anomalies because direct count of bacteria in samples are typically much higher than those of viable plate count only. So great plate anomalies only because many of them will not grow on the typical plate. And again, there will be many microbes that cannot grow without other microbes. And we will see later on when we talk about some methane-oxidizing organisms that really need to have some sulfur metabolism related organisms to live together. So they literally require each other. They live in symbiosis with each other and they can't be isolated one without each other. So they're fighting to each other. So you cannot cultivate them separately. So again, if you cannot cultivate organisms, does that mean that they're dead? Probably not. I mean, it can happen, but rarely. Maybe they are just non-culturable, or they are culturable, but you just did not find the right media for them. And in terms of growth phases, again, this is the picture that you probably saw multiple times, and it in this class will be something that I will always ask because I consider this general microbial culture. So this growth curve is in batch culture. Again, when we talk about continuous culture, which is in bioreactors, they have slightly different growth curves, and we will cover that later. What batch culture means stationary. So we simple prepare media, we inoculate bacteria, we give them food like carbon source. In case of E. coli, it's a glucose. In case of methanotroph, it's a methane. In case of ammonia oxidizers, it's carbon dioxide. So you don't add them anything else during the growth. You just culture them and you let them be. And then you observe those four growth of those four phases in bacterial growth. And they can be balanced or unbalanced. That's important. So the log phase is this flat line at the beginning. So this is something that in log phase, we never encounter growth rate. We will mention later growth rate. So log phase is a period of adjustment. So that's the moment when your bacteria is adjusting to the nutrients you gave them, and this is unbalanced growth. So that's why you don't count log phase in the growth rate, because there is no growth. Cells are just preparing. In exponential phase, or log phase, is the only balanced growth phase. So we call it log or exponential. So now all biosynthetic activities are like basically flourishing. So it's all great. Cells are growing, they're at maximum rate. But as they are exhausting nutrients, because remember, you're not giving them anything. You gave them one time, that's why it's batch culture, and they're now using available oxygen, they are aerobic, and they're growing and producing something. So in one moment, so up to one moment, they will produce metabolic compounds, but in one moment, they will exhaust all nutrients and they will start producing waste compounds that can often be excreted to the media. So that's when cells are coming into the stationary phase, which is this another flat line, which we say the number of live cells matches the number of dead cells. So what we say for exponential phase is that the number of living cells is higher than the number of non-living cells, while in the stationary phase, those two numbers are equal. In case of that phase, cells grow long enough, and then many toxins will be exhausted, will be excreted, and cell will stop growing. Now, lag and stationary phase are known as unbalanced growth phase, and in these phases, one or more aspects of bacterial growth and division is not timed with each other, so that's why they are unbalanced. So in lag phase, again, the first unbalanced growth phase, cells are still adjusting, so you still have synthesis of all molecules, metabolites required to complete division, growth. Literally, many things are happening, but not invisible. So it's still stable, it's still flat line. So cell machinery is not threatened. In another phase of unbalanced growth, so nutrients are not fully exhausted. So only partial nutrients are exhausted. So even though cell machinery is ready, there are no enough nutrients to continue growth. And if you would have continuous culture, which we will cover in the next slide, you would add to cells more nutrients so they can keep their machinery running. But the bottom line is that in both cases, the amount of nutrient is not timed with cell machinery, and that's why we call those two phases unbalanced phases. Another example of unbalanced growth, and there will be a bunch of scenarios or scripts that I will give you as examples, and I will post that either on e-class or we will share it later. When cell needs to readjust, when you move them from rich media like LP, where everything is present, they don't have to put any energy to make any nutrients, and you move them to minimal media, for example, where they have only available glucose and they have to synthesize all amino acids and everything. So that's where it's like a switch from rich to minimal media. So again, so this was all for unbalanced growth. In terms of log exponential phase, so this is where we count that growth rate. And you will see often in microbiology paper growth rate, but make sure you understand that growth rate can be counted only in log phase. So if you want to count the growth rate, you will have to take more time points. You will have to do it 0 hours, 12 hours, 36, well, 24, 36, but you have to have multiple points. It cannot be like 0 and then 48. So you have to monitor growth. And you only count the time period in between that. So you don't count this log phase. That doesn't matter in this case. So, and this is the formula. Of course, please don't memorize this formula. It's all everywhere. You can type it. You can find it wherever. I also don't know it, and I will never ask you to know. But basically what matters is to know what is the OD or number of cells at the end of log phase, at the beginning of log phase, and you need to know in between which time points is that log phase. So again, that balanced growth happens in log phase, but unbalanced growth is moment when one or more aspects of growth and division is not timed with each other. Log phase, mathematically determined. Please don't memorize the formula, but know how to apply this formula, and this is what I will show you. So what I will show you on this graph. So again, we use OD as a representation of number of cells. In ideal case, you would count cell number. So what you do, you look at growth. So you do OD, you determine where is the log phase, where is that slope, like log, and you determine in between which time points that happen, and that's NT minus N0, which is OD at the end and the beginning, and you need to know natural logarithm or logarithm of 10. And the generation time is, as I said at the beginning, time needed for doubling to occur. And growth rate constant, or n, is inverse of generation time, or 1 to 3. So how will you determine generation time, or how many minutes or hours you need for cells to double? So for E. coli, it's usually 20 minutes. For metanotrophs, it's 12 hours, but it really depends. So what you need to do is to observe the doubling absorbance at y-axis, which is here. So doubling absorbance is like 0.1 to 0.2. So that's where doubling happened, because from 0.1 times 2 is 0.2. And then you look at the x-axis where OD occurred. So the beginning occurred at 68 minutes. So 0.1 was at 68 minutes, and then doubling occurred at 110 minutes. So 110 minus 68 is 42 minutes. So 42 minutes is literally needed for this bacteria to double. So that's doubling time. And again, just a small practice that I can give more, but this is not, I believe that you all know how to do this. So I don't like exponential numbers, so I always convert them to the whole numbers. So if you start your culture with 300,000, 3 times 10 to the 5th cells, or 300,000 cells, how long it will take, like how many minutes, days, or hours will take for cell to reach 1.2 times 10 to the 6th, or 1,200,000 cells, if generation time is 20 minutes. So again, your starting cell number is this, 300,000. Final number is 1,200,000. One doubling will give you how many? Like 600,000 cells. And to reach 1,200,000 cells, you would have to have 2 doublings. So n is equal to, which means that your generation time is 40 minutes. And this would be all for this lecture. Again, I thought I will have time to start another lecture, but I won't, which is okay. We will finish next time.